Hi! Thank you for coming to this week's installment of the Zoom meeting, which someone in our own department said they didn't know that it was the academic meeting of the department every year. That's what the new meeting is. That's what this is. This week's meeting is being hosted by the Division of Ecology and Biostatistics, and I'm Landon, the head of division. And we're not in our usual venue, so thank you all for coming to this special venue. Particular people will filter in because they're still finding the place, I'm sure. Our speaker this week is Professor Mike Emch. This is Mike. Mike is the Distinguished Professor of Geography and Epidemiology at the University of North Carolina at Chapel Hill. He's been a friend of the department for about 5 or 6 years. He's been passing through Cape Town and giving a
Thank you.
Mike is, he doesn't like when I say this, so that's why I say it. Mike is the father of health geography in the United States. He's really one of the people involved in founding the discipline of spatial thinking around human health. And his graduates over, you're not that old, but his graduates over time have seeded out and started departments across the United States and indeed around the world. He does a huge amount of work in particular around spatial thinking in epidemiology and infectious diseases epidemiology in particular. It cuts across gastrointestinal diseases. We were talking about Shigella a moment ago, cholera work, and respiratory diseases, pneumonia, influenza, and so forth, and increasingly malaria and tuberculosis. And most recently, in fact, you're on this trip visiting Malawi, where he has a project site and is involved in some of you will have learned about the new RPSS vaccine, the new malaria vaccines that are coming out in October, where he's been very involved in some of the spatial analyses, which is so relevant, of course, to understanding malaria epidemiology in particular. Mike will be speaking to us today about spatial epidemiology and landscape genetics in 2 infectious conditions, malaria and influenza. I'm excited to say that we're going to see data I just discovered that is hot off the press. One of his postdocs sent data last night for the presentation today. So you'll be seeing things that no human has seen before, which is tremendously exciting. He's a great speaker, very affable. We're going to hold questions until the end. Mike will run for about 40, 45 minutes, 15 minutes of Q&A, and we can spill over if we need to. So Mike, I'm going to hand over to you with a huge thanks for joining us. It's always wonderful to have you with us, and we always learn from having you speak to our department. Over to you. Some way this better will work. I press those. The cameras should turn on. Yes, good.
There we go.
Thanks, Mike.
Thank you. I feel I'm like really high now. So anyways. So thanks for coming today. It's always great to come back to Cape Town. Landon asked that I talk a bit about spatial epidemiology, and I decided nowadays I seem to I don't really like to talk about the most polished of my topics. I like to talk about what I'm doing right now. So I'm going to give you some examples of what spatial epidemiology is and some of the methods and the applications we've done over the years. And then I'm going to talk about 2 studies that are ongoing. And one is on the RTSS malaria vaccine, and the other is on some influenza sort of etiology work. So spatial epidemiology, what is it? I've heard that there's quite a few of you here that are infectious disease epidemiologists that are in that class at least. And I mean, the definitions are simple. It's simply the subfield of epidemiologists focused on spatial distributions of health. And as a health geographer and spatial epidemiologist, we use spatially referenced data to look at spatial patterns and try to understand the processes that are producing those patterns. So I'm going to particularly talk today about the landscape genetics, about incorporating sort of genetic characterization of pathogens as well as their spatial distributions. So combining spatial and molecular epidemiology approaches. So the whole world and everybody in the scientific world knows about, have seen sort of, maps of COVID, and this is just something that I've pulled off of GISAID and looking at the sort of phylogenetic tree of COVID, and you can see the Omicron branch started here in South Africa, and then mapping the distribution on the right, and this is just literally like a screen capture on the GISAID site. And so you can look at this for a very long time and try to make sense of it, and then you can look at time series, and it's I have fun on websites like that. But this is the this is kind of the root of landscape genetics, is trying to understand the spatial distributions of genetics of pathogens, and landscape genetics, and I'll define it now, is a field that it's premised on the idea that they're exploring spatial variation of genetics and how organisms exist in and move across landscapes. And so again, pattern and process. There's two steps to a genetics landscape. Landscape genetics analysis, and that is, first is pattern and how genetic characteristics vary in space, and the second is process, and correlate those patterns with characteristics of the underlying landscape to try to understand why those genetic distributions are the way they are. And so that's the goal, is to try to understand landscape factors that drive evolutionary processes. This is very Darwinian. So this field has primarily, I assume most of you were health scientists, maybe you have previous lives in other kinds of biological sciences or whatever, but this field was developed primarily focused on plants and animals rather than pathogens and people, to do things like inferring colonization patterns, habitat restriction, extinction events in mammals, reptiles, insects, and plants. And then there's a more recent recognition that landscape genetic techniques can explore drivers of disease and spread of parasite transmission, gene flow of human illness, and things like that. So for infectious diseases, we can answer questions about what features of the human natural landscapes drive the emergence of evolution of pathogens. Questions we can ask: what local population environmental variables are related to the molecular evolution of a pathogen, or act as barriers to the spread of a pathogen, or drive the diffusion of a new variant like Omicron, right? Or what's the impact of vaccination campaigns on the evolution of that pathogen? There's we're in a big data era, both with spatial data. We all have our GPS devices in our pockets now, and then we've got sort of molecular techniques like PCR that have been developed. And so we've come a long way since John Snow's 1854 map of cholera, and then we can make more sophisticated maps. That's a map of malaria in the Congo that was derived from leftover blood spots from the DHS survey. That's 2013. We've done it in different years. So these methods allow us to sort of do some sophisticated work both on the spatial and the molecular side of things. So anyways, this is these are blood spots that are taken from the DHS survey in the Congo. We have leftover blood spots for HIV testing, if you don't know about DHS surveys, but they're population representative samples for countries and in low and middle income countries, and so that should say 2006, not 2007, and 2013, 14, and so so these are the data that I'll show you a couple examples of, and so we have all these technologies at our disposal. You know, this is one GPS device, and we have software and sort of, you know, code out there, our code, and everything that allows us to do spatial analysis. It's all of this open source data sets are just as good as the proprietary ones at this point. But we have all these gadgets as well that allow us to sort of, you know, collect data easily and cheaply. And so this is why we're in this sort of big data era for spatial data. But we've got incredible history of theory out there. These are just a few people that were sort of some of the leaders of this field of sort of spatial epidemiology and health geography, and so, you know, I always tell my students, when you have an idea, you should read, and pretty much everything's been digitized at this point. But at some point, I was telling them, go to the stacks of the libraries, because not everything's been digitized. But there's incredible theory out there, and oftentimes it's nothing to do with health, right? So Waldo Tobler, the one in the middle, there's something called the first law of geography, Tobler's first law of geography, and he died a couple of years ago. But and it's the things that are everything's related in space, but things that are closer together are more related. Okay, so it's a simple thing, anyways. But he did stuff on plant morphology, kind of like Darwin did with the finches in the Galapagos Islands, right? So and you know, and it's sort of like isolation by distance, which is the Darwinian concept that things are genetically more similar to when they're closer together, and when they're further apart, they're less related. And Darwin did that with sort of beak morphology of birds, but now we can look at, you know, sequences, like full sequences. Okay, so this is sort of one, like using those leftover blood spots from the DHS. This is one thing that we've done. We, you know, so I work with a whole bunch of molecular epidemiologists and a lot of people that are sort of good at, you know, at sequencing, you know, malaria parasites, and so you can look for these sort of genetics, these resistance markers in the pathogen, and so that is a map of SP resistant malaria in the Congo in 2013, and it's coming in from the east. I was in Congo just a few weeks ago, and this is a new study that UNC, my university, and our group is launching, that is actually looking at, you know, artemisinin resistance, and there's a lot of emergence in places to the east, like Rwanda and Uganda, and it's coming into the Congo, and so this project is basically to develop the capacity and have all these sites, 35 sites around the country, and anyways, and so this is one example of how we, and so sort of understanding the flow in space and sort of gene flow is the goal of that project. Another one, called it stealth malaria. So almost all malaria tests are rapid diagnostic tests, right? It's like a COVID test, you know, but blood, and rather than people put stuff down your throat. Anyways, and so these they it's targeting a gene, HRP2, and and these parasites are tricky, and they evolve to evade detection of this RDT sometimes, right? And so what we did was we took those leftover blood spots, and we looked for deletions in that HRP2 gene, and which can cause a false negative of RDT results, which is really problematic because test and treat is one of the main types of interventions for malaria, and so this is a problem with these mutants, the serious problems with malaria elimination, and so this is just one data set. We just a small data set that we looked at, and there were 6% of the in the Congo, but you could had these HRP2 deleted. They were HRP2 deleted parasites. And then you look at the map, and there have been sort of more local level analysis that's looked at sort of tried to understand the drivers of this, but this is just a course level view. And it's spatially heterogeneous. It's clustered, right? And it's clustered in like North and South Kivu and over in Kinshasa. And anyways, and so from a programmatic standpoint, this is where the problems are for test and treat, but we can also try to understand why. We can also try to understand why what's driving these processes. Okay, so those are just kind of like to get you thinking about these, and now I'm going to talk about influenza research, and this is ongoing. Quite a few years ago, I guess 15 now at least, I started working with a guy named Shifeng Wan, Henry, he goes by. He's in the US. He's originally from China, but he's the first one to identify H5N1 bird flu in Guangzhou in China. And he's a professor in the States now. And we worked on H5N1, but I'll give you a little background on how many people know about flu here, a little bit about it, we've all had it. So, influenza viruses cross species barriers, and, with close contacts with birds, in particular domestic poultry. It's resulted in interspecies transmission of influenza to non-avian hosts, and some transmission of influenza is from wild migratory birds to domestic or captive mammals. There was my work on H5N1 with Henry and others was in China, but also in Vietnam. There was a major outbreak of H5N1 in 2003, and the source was the introduction of poultry trade across the Chinese border. Chickens were cheaper on the Chinese side, so they came across the border even though it was illegal. And so the factors that were driving that H5N1 in Vietnam were this shared border with China. People have like a cultural preference for live or freshly killed poultry. There's this ecosystem, and I'll show you a little graphic of that ecosystem. So poultry is integral to agricultural production in rural Vietnam. Everybody's got chickens in their backyard, or maybe less so now as the country has gotten richer. But and 80% at the time had backyard poultry flocks. So some people in my field of health geography called me a disease ecologist. I guess I would call myself a disease ecologist as well. And some of my colleagues, they're sort of on the mathematical biological disease ecology field, they oftentimes don't incorporate humans into the ecosystem as we do within the field of geography. So humans are integral parts of this ecosystem as well as the environment. I guess you could call this a One Health project. We studied elements in the social and natural environments that drive Vietnamese landscape, of the Vietnamese landscape that are driving the genetic variation of H5N1, not just the disease, right, in chickens or ducks or humans, which we looked at all, but also the molecular change, the genetic change. So it's an anthropozoonotic pathogen that and the majority of infected birds in Vietnam are domesticated birds, chickens and ducks, and humans interact with these chickens and ducks. They're part of the family, if you will. The goal is to understand molecular change as an outcome of interacting environmental and social pressures hypothesized to drive that molecular change. So you can see in that little sort of graphic that's in this paper by my former PhD student at University of Iowa, Maggie Carroll. And a lot of these are about the human side of things. Some of them are about the environmental side, but they're about things like population density and the socioeconomic status, like socioeconomic status comes into play, because if you're rich and there's a bird that is a little sick, you're not going to eat it. But if you're poor, you're probably going to eat it. So this is a busy graph, and I'm going to go back to Darwin here and the concept of isolation by distance. If we look at in this room, all of you are a point in a distribution, and so we could take any variable and say the people that are next to one another, are you more similar, and how in whatever variable it might be. It might be by your major or your specialization, which is, there's probably clustering of a lot of things in this room because you're hanging out with your friends, right? Well, and we could actually quantify that distribution and create something called a correlogram, which would be like, how related are things in space to one another? Okay, how related is this variable? So we could do that with wealth. Maybe the rich people in this room are hanging out with the rich people, and the poorer folks are hanging out with poorer folks. So we could quantify that, and I'm sure that we would probably find some kind of relationship. I don't know about that one, but it's something. So anyways, we can actually, and we can. That's a big distribution there, and then that is separated by year and by gene. But these are the correlograms, the Mentel correlograms, and what these show is, this is the spatial distance. Vietnam's a long country, it's 1,500 kilometers, and this is how correlated the different observations are in space. Okay, and what we found, and we wrote a lot of papers and they're listed here. I'm happy to give you the and so so evolution of the H5N1 viruses and most of those data are poultry, is highly correlated with location and the spread of those in geographic space. There was a sort of classic isolation by distance pattern, but not only, right? there were other variables driving that. Things like population density and as well as population density as well as sort of socioeconomic variables as well. Okay, there's this is all about sort of this ecosystem. There's a combination of suitable environments for species mixing, you know, of humans and birds and different birds with one another, and the presence of you know, of high number of hosts. So an animal market might be one of those places, or you know, when humans are involved, sort of human interaction. You know, you think about the Wuhan, you know, Hunan market, the animal market and COVID. You know, that story. If you haven't read the science papers on that, there's one molecular paper and one spatial paper. Read them; they're interesting papers. But so yeah, okay. So that's kind of the old work that I've done for a long time on H5N1, and this has moved to a new project on swine flu. Pigs are a great mixing vessel for flu viruses, right? Humans, birds, and pigs, and different kinds of mammals. One of my collaborators on this project is a guy named Richard Webby, and his advisor is Robert Webster, who was the person that sort of in the, I think it was in the 1960s or 70s, was like, you know, finding flu in all these different mammals, including whales. It's like putting like swabs down their blowholes and things like that. But and he's still at St. Jude in Memphis. He's in his 90s now. And so anyways, we have we can this graphic is basically just showing you that all of these flu pandemics, the four in the last 100 years, have been they're related to one another. Okay, there's genetic there are sort of, you know, parts of the sequences are related to one another. The H1N1 in 1918, which was, you know, the Spanish flu, which started in the United States, and then the 2009 flu. There's gene segments that are the same. Okay, and so our study right now that's ongoing is we're looking at novel swine influenza viruses, and we're looking at you know, how they continue to emerge and evolve and to be highly transmissible viruses. And then these are the hypotheses, and then there's intercontinental movement of gene segments in the US and China, which is where the study is, that facilitate the emergence of novel swine influenza viruses. We're actually looking for reassortment events. That's what we're doing. We're collecting lots and lots of viruses. And then that last one is really the landscape genetics one. And that is that there's a unique set of ecological factors that's driving the evolutionary dynamics of these influenza A viruses. And the two largest swine industries are in the US and China. China is bigger than the US. Basically, it's like a Ford motor plant. It's like, you know, 6 months life cycle of these pigs, and they're massive, and we bake bacon, you know, at the end of it. And in China, it's mostly mechanized at this point as well. So, okay, this is a really busy graph, but basically this is my Richard Webby and this vet professor at the University of Illinois, Jim Lowe, who knows more about pigs than anybody in the world, I think. And so this is they move 3 times in their lives, right? They're born at one farm, then they sort of spend their adolescent months in another farm, and then the last farm is they fatten them up, and then they make bacon out of it. Okay, and 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 so you know, this is I wasn't involved in this paper, but Richard and Jim did it, and so those are, you know, influenza cases over time, and these are all the words that define the different stages of, like, when they're babies and things like this. These are different pig words, and then these different colors are subtypes of influenza, and so you can, you know, like H1N1, H1N2, right? And you can look at these, and you can, you know, you can look at this for a long time, but you can look at there's clustering of the types of you know, within the age structure of the pigs, as well as the over time. Okay, so those aren't those aren't spatially random graphics. Okay, there's another one, and there's a guy named Tom DeLiberto who's another collaborator in this project, and he's the keeper of. We have wild pigs in the United States. Are there any wild pigs here in South Africa? No. Well, they warthogs. Yeah. Right. It's always, they seem. I went to Namibia once, and they were always in the road. But anyways, so that really nobody knows if there's gene flow from feral pigs to the domestic pigs, right? And these things are nasty. They get into the farms and things like, even these big companies, right? They get into there. So Tom he's the sort of the head of animal health for the U.S. Department of Agriculture, and the keeper of all the feral swine data. And we're actually in the virus data. So we're actually collecting flu sequences from them as well. So on the China side, this is our collaborator on the China side wrote this paper, and there was this sort of newly emerged genotype or reassortment of an H1N1 virus, and humans, the swine workers, 10% of them actually had this virus. We're not studying that in the United States. It's a little hard to get the swine industry to work with us on that one, but anyways, this greatly increases the opportunity for adaptation in humans, and raises concern for a pandemic, virus. Okay, so this is the postdoc I was talking to last night. His name's Varun Goel, and that's just a picture of a pig. And Varun, that's actually not my picture. That's from a science paper. It's just a nice picture of a pig. And Varun did that. And we have virus sampling going on, and these are the different places we have them in the United States, in the Midwest, many states, including Illinois, but also in multiple places in China. That's where the sampling sites. The swine industry, I told you, they live in 3 different farms. Sometimes they're within different companies, and A, B, C, D, and E are all the different companies. Okay, and those are those swine farming systems they're supposed to be biosecure, and there's not supposed to be gene flow between one farming system and another farming system, but there probably is. That's what our hypothesis is. Okay, and so sometimes sometimes the farm's like right next door, and sometimes they have to truck them 100 miles. Okay. So we are collecting lots of different virus sequences from all of these places. You'll recognize, this clade classification system, Delta, Omicron, things like that, right? And there's some characteristics of these different sort of clades of that for swine influenza, and some of them, you know, are sort of this, you know, seasonal flu. Some of them are pandemic flus and things like this. The NPDM one is one that is associated with the 2009 H1N1 pandemic. These distributions of the different clades in this is our data, have changed over time and in space. Okay, I'm not going to go through each of these because I want to get to my other example too, but we sort of mapped out the different distributions of the different clades over time, and some of them appear and disappear, and some of them are persistent. Okay, and so our question, one of our questions, we've got a lot of questions, but one of our questions is there the clustering of the virus just within the different farm companies and systems, or do they cross into different systems because they're supposed to be biosecure, right? And if there's gene flow between the different systems, is there a classic isolation by distance pattern? And the reality is that there's a lot of clustering within systems, but there's also isolation by distance. Okay, these are the ones that these slides were produced last night, which was yesterday morning in the States, and yesterday afternoon, and these this was done in bees. If anybody is a bioinformatics person, but these are the different sort of clades for the data sets, and these are multiple Midwestern, like Illinois and Iowa, and anyways, and these are the different clades, and NPDM is one that is persistent and highly prevalent over time. Okay, this is another, you know, this you can, based on sort of you know, the sequence data, you can actually look at the directionality of, you know, which way the gene flow is going. And there's this one company, in that first slide I had A, B, C, D, and E, and we should probably do that here because this kind of implicates ESHS. I don't even know what that stands for. It's one of the companies, but it's the source of transmission to the other companies, right? And anyways, and so, you know, we have data dating back to 2012, and that's sort of the direction. It's you know, you could look at these graphics for a while, and you could say it's much more complicated than that. And then sometimes it you know, goes to another system, and then it goes to another system. But yeah, so they're not closed systems. This is a very busy graph, which are basically the phylogenetic trees, that, you know, that basically show you know, the different branches from the different systems, and this is by system, which is the colors there. So anyways, so this is ongoing. What we're doing now is there's a whole bunch of ecological covariates that we're looking at in these farming systems. We have pen level data, we've got barn level, farm level data, pig flow from one farm to the other, and then we have, you know, all kinds of gadgets that are collecting data because this is a Ford Motor plant of pigs, and so they have to keep the temperature just right and things like that. And so we have all these data, and some of them might be environmental data that's driving it. And then we have sort of a fancy sort of machine learning group that is actually trying to make sense out of all of these variables as well. So that's what we're doing, and again, the major goal with this sort of deep learning methods is to figure out what one of the reassortments like what drives the reassortment and what drives a persistent reassortment, the one that's going to stick, because there's reassortment, we find reassortments all the time, sort of, but which ones are going to be sticking? And if we can do that, wow, that would be great. This is not easy though. And so, okay, I'll try to do this in five or so minutes, just so we have time to talk about it. But this is this ongoing RTSS malaria vaccine trial. It was done in a, I'm not going to show you that. This is the last talk that I gave at UCT a few years ago on cholera, but and the work now on RTSS built from that. But anyways, probably none of you were at that lecture a few years ago. But anyways, this big trial, 11 sites, 7 countries. We at UNC did the phase 3 trial site in Malawi and Lilongwe, but in this analysis, we have data from 3 different countries in Lilongwe, in Kintampo, Ghana, which is sort of central Ghana, and Lambaréné, Ghana, central Gabon. And we chose those because they're ecologically very different from one another. Okay, this is one of these moderate to low moderate efficacy vaccines, but it's the only one out there. Although a couple of countries licensed this R21 vaccine recently, but as well, but basically it's the only one. And so the overall efficacy was only 36%, but it varied widely from down, in Mozambique it was 22% up to 75% in Kenya, and it was about 50% in Malawi. Anyways, and so we are trying to figure out why, why efficacy is different in different places. Okay, and there's 3 arms to this study, and one is sort of the ecological arm, is things like transmission intensity and the things that drive transmission intensity. Okay, and then so like Congo, like 30% of the country right now has malaria, right? Of the 80 million people. In South Africa, maybe there's a few cases along the border in Zim and in Mozambique, but not many, right? And probably not this time of year. But anyways, so sort of ecological factors, and then the other is host genetic factors, human host genetic factors like HLA, things like that. And the other is parasite genetics. Okay, so these are the 3 things that are probably, ecology, host, and parasite genetics are probably driving differences in efficacy. These are just examples of the different ecological variables that we as geographers measure, and some of them are obviously like questionnaire, like age and sex and things like that. Some malaria ecology, like whether you're using insecticide to treated nets, and this is just some images of the 3, and we can look at things that vary over space and vary over time. So this is in our Lilongwe site. This is bed net use over time, and 53% in the beginning of the trial, and up to 80%. And then this is, and we can also look at individual bed net use, but if your neighbors are using bed nets, that's good for you too, because you get this herd effect of bed net use, right? And so we could measure bed net use not just individually through a questionnaire, but also spatially. And malaria prevalence went down over time, and there weren't that many people vaccinated, so it really wasn't the vaccine that was doing it. So let's just say there's a lot of parasite diversity, trust me on that. And there's even human host genetic diversity, even within Malawi, but definitely within the 3 countries. Okay, so I'll show you just 2 more slides on this. The purple here is all the ecological factors. The blue is human host, and then the yellow is parasite genetics, and this is a paper that we published a couple years ago or maybe a year ago. And so that just shows the different doses that people are getting in the vaccine response, and then this one here, that's the booster. Okay, you can see that the vaccine protection goes way up after the booster. Okay, and then it starts to wane. Okay, and then there's this is data in those 3 countries, and when you're getting a disease like malaria, then you have an immune system and you're developing antibodies, and then you're protected, so you can have natural immunity. We all had COVID, and I got every vaccine that I could get in COVID too, and so and so we know this story personally, even if you're not a health scientist, and so the people who were not vaccinated have some natural protection, and it begins to wane over time. But at some point, the naturally protected people had a higher protection than the folks who got the vaccine. Now, this is not an argument that we shouldn't have a vaccine, by the way. So I want you to read that paper if that came to your. This is an argument for when another boost and also a timing of a boost. But I'm going to finish here rather than going. This is another paper that we had antibodies, and so we actually measured it not just in disease outcome, but sort of in antibody titers. So there's some growing evidence, both genetic variation in the parasite and variation in human host genetic factors affect RTSS efficacy, and that genetic factors may be interacting in complex ways to produce variation in the natural and the vaccine-induced immune responses that protect against malaria. And transmission intensity, so how many people in an area, how much transmission is in an area, has a role in pre and post vaccination periods in sort of modulating immune response and sort of efficacy of the vaccine as well. So I know that was a whirlwind tour. I just wanted to, as Landon asked me to give a taste of what we do in spatial epidemiology. Those were tidbits of examples. I'm happy to talk about any other types of spatial analyses that can be useful in health studies. These are I've got a little group called the Spatial Health Research Group at my university, and then we have a larger group called IDEAL, which is the Infectious Disease Epidemiology and Ecology Lab that is basically a combination of molecular and spatial epidemiologists at UNC, the people that gave us money. That's it. Thank you.
Mike, thank you for opening your eyes and giving us a taste. It's a taste of conditions that are critical to human health from there in terms of that we don't in this department spend a huge amount of time for this somewhere, but gives a taste of that, but also a taste of what spatial epidemiologists do and how they think. Thinking about distributions of things, but with the distributions in the influenza example, for instance, is genetic diversity of the pathogen rather than necessarily be infected or uninfected. Kind of the places to think to, and at the minimum, we all know where we're going to get food from next time. Stay away from pigs. That's one of my takeaways. Let's open up for, I think we probably have max of 10 minutes for Q&A. If you have any questions for Mike about malaria diversity, influenza diversity, thinking about how we think about the spatial distributions of pathogens and of health and disease states in populations.
Yes.
Thompson.
So I know that the vaccine, the SPH, is made of recombinant protein plasmodium vector, and it's also mixed with a protein from hepatitis B. So when you said efficacy different in different populations, do you also get the immune response from those different populations based on hepatitis B on that protein?
I always get these questions, and then and my colleague Jeff Brown, Jeff Bailey at Brown could answer your sort of immunology question. Good question, and I don't know. Is the real answer. It's a
good question. Tom's probably not for the geographer, but for the immunologist.
yeah. So yeah. Yeah, it. So, I'm gonna show you the slide that I didn't show you on cholera, just to answer that question. So my colleague John Clements, who and Muhammad Ali, we made an observation 20 years ago that a typhoid vaccine had 3 different trials for the same vaccine in 3 different countries: Chile, Egypt, and Indonesia, and the efficacy was 10%, 50%, and 90%. And it's like, why? And the answer was, it's got to be something about those places. So this oral cholera vaccine, had been around for a long time, and it wasn't being used, and a lot of people, including John, who's older than me, and my other colleagues, one named David Sack, and other people, were advocating for this vaccine to be produced and stockpiled by WHO, and it wasn't. So there's a whole bunch of papers, and they basically have the same result, which is that there was this big herd effect that really you got more bang for your buck out of this vaccine. So if this side of the room, if 90% of you are vaccinated, and the 10% just aren't going to get it. Actually, anything over 50% was going to be fine, right? And then over here, got a lot of people who don't want to get vaccinated, and then so the people that whatever, there was a herd effect, and there were some other things about sort of the local ecology as well, that were driving whether this vaccine worked. And this is in Bangladesh, in a population of a couple 100,000 people, in a place called Matlab, Bangladesh, which is a very famous health research site. So back to malaria. So why? There's certainly parasite genetic differences that we know about and have already published on, that is driving part. The vaccine worked better against some parasites than others. Okay, that's part of it. But transmission intensity is important as well. Then you can actually get into something more detailed about transmission intensity as well. It's probably more affected in the higher transmission intensity areas, but the devil's in the details. So in our group, we have people that are really good at this environmental measurement, and so that's kind of why. I mean, we're not studying Mozambique and Kenya. We actually want to further this study after the one that we're doing right now and do it in all 11 sites, or as many as we can get data for. So I don't know if that answered your question, but you know, there's something about these places, and we don't know about the human host genetics as well. That's Jeff is doing that at Brown University. So the parasite, the blood from the kids that were in the trial is in Rhode Island now. He's trying to sort out some technical difficulties, but yeah.
So Mike, if I hear what you're saying, so your question, it's not something you would adjust for per se. It's that the settings are fundamentally different, and thus the transmissions.
Yeah.
We have time for one more question.
Somebody want to ask me about cholera?
No, but I want to ask no one else does, and I thought this is like, how do you think about interventions in spatial epidemiology and health? Because what you presented to us is critically important, it's fascinating, but it's really describing in great detail and understanding the nature of the problems. So take the swine flu example. Is the intervention, now I want to go see what VHS is. I'm going to go Google that. But is the intervention to shut down that, or to intervene on the location?
You know, for swine flu in particular, or for any?
Or for anything. How do you think about intervention?
So there's actually a book that is kind of like we wrote one chapter of the book. It's about sort of the geography of interventions, and so you know, economists think about it that this is like impact evaluation, and RCTs are one kind of, you know, evaluation of something, and what RCTs don't typically do is incorporate some contextual effects or sort of place effects. They generally look at individual level effects, sort of in an RCT, right? And so the place effects might be just as important for some outcomes as others. And so that's kind of what we did with cholera, and is what we're doing with malaria, is we're actually incorporating place effects into like a reanalysis of the original protocol of the vaccine. There's some people around here that are involved with TB vaccines, and we're doing one. We're one of the 30 sites of this new Gates TB vaccine, and there's, you know, there's clearly going to be some place effects of TB. Obviously, the transmission intensity of TB in Cape Town, let alone of all of South Africa, is, you know, if you look at, you know, the Cape Flats versus, you know, someone right around here, I mean, they're obviously different. So those place effects are really important to incorporate in some, and sort of back to that slide on that theory, like theorizing why and the mechanisms for which those place effects might change, you know, modify the effect of whatever it is that you're measuring. And that's what these trials tend to not do. After we did the cholera vaccine trial, we were looking for a vaccine to do this work in a prospective, you know, John was the PI of the original cholera vaccine, but it was a reanalysis of that data after the fact. But then we wanted to do it prospectively with the malaria vaccine. So we built in, at least in the Malawi site, we built in some other data collection about ecological variation. So I don't know if that answered your question, but place effects are, if you get nothing from what, at least I'm in my soapbox, is place effects are really important, and for people that haven't thought about place effects. If you're a molecular person, you might not have thought about it, but they're important.
It's a good note to end on. Mike, thank you again very much. Thank you all for attending.